So today we're going to be going through a number of different sections and sharing with you overall about this data and how to apply for access. There'll be 4 different sections that we'll be sharing with you. So first, we're going to start off with Mark Effingham, the Deputy CEO of UK Biobank, and he'll be giving you an overview of the UK Biobank and data. We'll then turn to our Regeneron Genetics Center colleagues, Will Salerno and Sheldon Bai, who'll be taking us through the actual 300K whole exome data, giving you an overview and best practices for that data. We'll then move to Lauren Carson, Head of Research Development at UK Biobank, who will talk to you about how to access and how to apply to use the resource at UK Biobank. And then we'll wrap up with Anastasia Zelikova, getting on to wrap, how to sign up, create a project, and also data organization from DNA Nexus. Once we get started, if you have any questions, feel free to type them into the Q&A. Once the sessions kick off, in between, we will have Q&A sessions, and Caroline Clark, the Senior Project and PMO Manager from UK Biobank, will manage those Q&A sessions in between each of the sessions. If we have any additional Q&A sessions or additional Q&A that aren't able to be answered, we'll then look to move them over into our UK Biobank DNA Nexus community forum so that we can address those questions in that forum. And with that, I'll kick it over to Mark Effingham. Thanks, Mark.
Excellent. Thanks, Asha, and delighted to have the opportunity to talk to this group today in this webinar. And good to see, actually, there's a reasonable number of attendees who aren't actually yet biobank kind of researchers. And so we thought it'd be useful just to start off with a brief overview of biobank for those less familiar, and to give a sense of some of the data that is available in the resource for research that's in the good of public health. So without further ado, in terms of just a bit of context about biobank for those who perhaps are less familiar with it, it's a prospective research study that recruited 500,000 people from all across the UK between 2006 and 2010. The people at time of recruitment were aged between 40 and 69. The majority came across from England, but we've got good participation from Scotland and Wales. And it's an entirely altruistic endeavour in that those participants have joined the study. They don't get any compensation. They don't get any feedback of results. It is really about creating a resource to help inform health research for future generations. Now, those participants came to one of a number of assessment centres that were located across the UK to get a good representation from across the country. And they spent probably about an hour and a half with us at the time to provide information about themselves through the touchscreen questionnaires. So answering questions about lifestyle and environmental exposures, about family health history, the number of cognitive tests and hearing tests, for example, and included a number of physical measures such as height, weight, grip strength, spirometry. And they also provided a number of samples, which predominantly went into storage for future assays, so blood, urine, saliva. And importantly, they provided consent for us to be able to access their health-related records over time. It's clearly it's too expensive to bring people back to assessment centres, and we follow health outcomes through linkage to these records. They also provided the ability for us to recontact them to take part in study enhancements, such as web-based questionnaires or the imaging visits, which I'll touch on shortly. So since baseline collection, there has been a number of enhancement projects which have added further data to the resource, which is now available to researchers to access. So in terms of hematology and biochemistry assays that have been undertaken, at baseline, the only assay that was done was on a fresh blood sample for a set of hematology measures, and they've been available in the resource now for some time. All of the other samples collected at baseline went into storage for future assays. In terms of biochemistry markers, in the last few years, we've recently completed a project to measure 34 biomarkers from a number of different sample types. And these data are now available in the resource for researchers to use. And they span a number of measures which we know will be important to understanding progression in particular disease areas such as cardiovascular, cancer, etc. Interestingly, there are two current access-related projects which are adding additional biomarker measures. So a particular note is the Nightingale Health assay that's been undertaken on the whole cohort, which is measuring some 249 lipidomic metabolic markers. And we made available the first set of data for 120,000 participants earlier this year. There is also a project being undertaken by another industry consortium who are undertaking a set of proteomic measures using the Olink platform, and that's looking to measure around 1,500 blood-based proteomic biomarkers on an initial 50,000 participants. We hope that funding will be committed in due course for the whole cohort, but those initial data should be available to the research community in the latter half of next year. So beyond hematology and biochemistry markers, we undertake a set of regular web-based questionnaires. So we typically do 2 questionnaires a year, and we try to use these to get access to information about participants that we can't get from linked healthcare record data. So for example, we've done web-based questionnaires on food and nutrition, on working life history, on sleep. And again, we've got a roadmap of questionnaires spanning probably the next 2 or 3 years of future questionnaires that we will undertake. And we get really good response rates. They typically go out to participants for whom we have email addresses, and we tend to get around 200,000 completed questionnaires for each questionnaire we do. We've also undertaken a set of repeat assessments on 20,000 participants in 2013 and 2014. That was at our Cheadle-based assessment centre, but we're also doing repeat assessment visits as part of the imaging study, which I'll touch on shortly. We've also done activity monitoring, so this is kind of pre-Fitbit days. We sent out a wrist-worn accelerometry device to 100,000 participants to get a measure of activity across the cohort. We did that for 100,000 participants who wore one of these devices for a period of a week. We've worked with external groups to turn those raw data, which are available directly, but into summary activity measures which researchers can use. And we've gone back to a subset of that cohort to look at seasonal variation, where a proportion of participants have worn one of these devices 4 times a year to look at variation across the seasons. And again, all of those data, both the raw outputs plus the summary metrics, are available within the resource today. And over the last 2 or 3 years, there's been a lot of focus on the genetic characterization of the resource and adding genotype and sequencing data. And that started with genotyping of all 500,000 biobank participants, which was done after we undertook DNA extraction from one of the buffy coat samples that participants had provided. And we worked with an external group to build a custom genotyping array measuring 850,000 direct measures. And then imputation, again by a group externally, who've taken those direct measures and imputed a data set for about 90 million variants. And again, that's now been made available to researchers since 2017. Around half of all applications to Biobank request genetic data, and we're already seeing very large amounts of findings being published from use of these data, including associations to the outputs, and also the likes of polygenic risk scores, which have been developed off the back of the UK Biobank genotyping data, and which are now finding a way into frontline clinical care. Following genotyping, there was an industry-led consortium led by Regeneron, and I'll let Will talk to this in more detail in a moment. But that's been undertaking exome sequencing, measuring the coding region of the genome. We made the first 50,000 exomes available back in early 2019, and the most recent release was for 300,000 exomes last week, and Will talk about that in a moment. The next release will be for a further 150,000 exomes to take it to 450,000 in total, and that will be occurring around about the end of October. there'll be more information going on that in due course via the Access emailing list. But I'll let Will talk to this in more detail in a moment. Following on from whole exome sequencing, there is a separate consortium with both industry and government and charity funding that is undertaking whole genome sequencing for all 500,000 participants. That project is proceeding well despite the pandemic, and sequencing will complete around about the end of this year. Data for the first 200,000 genomes will be made available in November, and we'd expect the remaining data to complete the 500,000 cohort would be available in early 2023. And then in addition to genotype sequencing, there is a current large-scale program that's being run looking at adding imaging measures to the resource. And this is probably the largest study of its kind, where we have a target to invite back 100,000 biobank participants to undergo a series of comprehensive imaging measures, as well as a full repeat of the baseline baseline assessment centre visit. So we're about halfway through that programme, with about 50,000 participants having attended one of our 4 dedicated imaging centres, and we expect to conclude the study in 2023. Participants spend about 4.5 or 5 hours with us, so it's quite a long visit, where they undergo a series of comprehensive MRI measures of brain, cardiac and abdomen, plus carotid ultrasound and full DEXA, and as I say, a full repeat of the baseline assessment visit. Those data are available in the resource shortly after participants visit, so they're constantly streamed in. But we're also working with, again, an external community of researchers who are generating what we call image-derived phenotypes. So turning those raw imaging scans into measures that researchers can more easily use as covariates within their research, such as kind of brain volume measures and similar. And there's already around about 3,000 brain measures available today, and other measures now appearing in cardiac and other modalities. And excitingly, we have been able to confirm recently the funding has been committed to undertake a repeat imaging program, where we'll be inviting back up to 60,000 participants to come for a repeat imaging visit some years after the first visit, and that will allow researchers to start to look at change over time, which will be a really important and valuable addition to the resource in due course. That project will start in 2022 and will end around about 2028. So it's going to be a long-term program. And then lastly, just to pick up on how we follow the health of participants. As I say, it would be impractical and too costly to invite participants to come back to assessment centres to look at how their health outcomes develop. And so we do that by linkage through to health records. And in the UK, we are very well set up with this, with access to the NHS sources of data. So we bring in regular updates of death data, of cancer data with data from the Enhanced Cancer Registry, also data from hospitalisation records from secondary care, and also primary care. And our focus has been very much in the last couple of years on increasing the primary care data we have available on participants. So we have that on about 45% so far. During the COVID pandemic, we have been able to get more complete coverage of primary care records for participants for the purposes of COVID-related research. And our focus remains on collecting those data for broader research purposes downstream once we are through the pandemic. But with those records coming through, we can identify cases of disease. And here just shows a quick table showing that even today, with the number of observed cases and what we would predict in the next 5 and 10 years, the biobank study is already set up to be able to do good studies with cases controls in diseases of interest to the biobank research community. So hopefully that's just given a bit of a flavour about the kind of data that is available within the resource. What I suggest we do, because I know I want to make sure Will has some good time to focus on the Exome data release that was available last week. I suggest we park the Q&A for this question session to the end and perhaps hand over to Will at this point to talk about more detail on the 300K Exome release that was made last week. So Will, can I hand it over to you?
Yep, thanks so much, Mark. And thanks everyone for joining. I'm going to do a quick run through of the 300K Exome dataset that was released last week by UK Biobank on the RAP on behalf of the UK Biobank Exome Sequencing Consortium, which is Regeneron Genetics Center and all of our pharma partners. So really appreciate everyone's effort getting these together. If we can go to the next slide. So the 300K Exome dataset is just over 300,000 individuals, participants that include the bulk of the individuals that were in the previous Exome releases, the 200K and the 50K. There were, some number of withdrawals, and that's why the numbers aren't exact. For everyone who's new to our Exome data, at UK Biobank, it is just under a 40 megabase pair custom capture design. All the sequencing is done on the NovaSeq 6000, 75 by 75 base pair paired end reads. And all of the data is processed on what we've termed our OQFE protocol. I'm going to go into that in a moment. But I think the big takeaway here is that all of our exome data are processed on this protocol, which was harmonized with other UK Biobank sequencing efforts, including the whole genome sequencing. The idea being that we want people who are analyzing sequencing data in any form from the UK Biobank to be able to allow these data to interoperate. There's a couple of metrics there on the right just to observe. The total amount of data for this release is about 300 terabytes, which is sitting on the RAP. These data comprise CRAM files, sample level, which contains all of our reads, GVCFs, which contain all of our variants and their respective indices. And then as an aggregate, we have the variants in PVCF, Plink, and BGen format. And then there's some auxiliary files, which you can read about with our documentation. If you can go to the next slide. So I'm not going to go through this in a ton of detail because everyone can read and scan those links to all of the documentation. But our OQFE processing pipeline is something that we developed for UK Biobank. It was applied to the 200K data and it will be applied to the 450K data. It's using the GRCH38 reference in an all-to-where manner. There's a couple other details there about how we do our variant calling. But one thing I do want to emphasize is that these CRAM files that we process, these OQFE CRAMs, they contain all of the reads that we generate. And they contain the original quality scores. So the full FASTQ files, the full raw data can be recovered from these CRAMs. We aggregate these data with GLNexus, and then there's some post-aggregation processing we do in order to make these data as amenable to as broad a set of research goals as we can. We don't do excessive filtering. We don't try to refine the data for any specific purpose. We're really trying to provide the UK Biobank researchers everything that they might need to do any analysis they might imagine. And the process by which we did all of this is available in extensive documentation that's on the RAP. You could find this in the science corner on the RAP and through the showcase. And if there's any questions about the documentation, please direct them to the community forums and we'll be sure to address those. But the last thing I'll emphasize about this protocol is that it's all built on open source tools. These methods and protocols are all out there. You can scan the links on the right. And again, if you have questions, please don't hesitate to reach out. Before we go to the next slide, I will observe that we have gotten feedback on our previous releases about how would users best do certain filtering. So if you go onto the science corner, you will find documentation about suggested ways of doing batch covariates for association analyses and how to do some variant filtering that would be appropriate for those analyses and for these data. Really suggest that before you dig in and do any large scale analyses on these data, you do read through some of those best practices. And always keep in mind that we do provide this multi-sample PVCF that contains all of the aggregate information for the variants. So if you would like to do your own custom filtering, you have the opportunity to do so. If you pop to the next slide, I'll just end on a couple of summary metrics. In this data set, we've identified 21 million variants. That's both in our target capture region, which is designed to capture, it's an exome capture protein coding regions. We also call variants in the buffer region, 100 base pairs on either side of those targets, which brings us to the 21 million. Sheldon on the team has worked up a nice breakdown of the balance between the target and buffer, the SNPs and indels. We also have provided a list of variants that, based on feedback from the 200K, would potentially be low quality and lead to some spurious associations. There's just under 5 million of such variants. Most of them are in these buffer regions. But if you read our best practice documentation, you will see that filtering these out is probably the best way to avoid getting some of these spurious associations in the downstream analyses. So I know that was a lot of information to throw out. Again, there's a lot of documentation on the wrap and on the showcase that we encourage people to look through. And then, of course, the community forums will be an amazing resource where you can access both the RGC's expertise in putting these data together and the broader community's expertise in how to analyze these data. So again, really thank you for your time. Please throw those questions into the Q&A and we'll make sure we get to them as appropriate.
So perhaps, Will, we can take, if there's any Q&A coming through now, perhaps one question I might have, Will, for you is perhaps just touch on, with the 300K release made last week, how did that processing compare to the previous release to the 50K and the 200K? And how will it differ to, say, the 450K release later in October?
Thanks, Mark. So the 50K, the original release, was done on a different protocol, and based on feedback, we updated to the OQFE protocol, which was used on the 200K release, on this 300K, and will be used on the 450. So the next release that comes out will be on the exact same protocol, and people should expect all of those methods to be exactly the same as described in this documentation and what we presented here.
Well, there's a question coming through around where will the best practices documentation be made available? I think you've touched on this already. I think it will be made available in the platform, I think within the Science Corner. It's also available as a resource within the UK Biobank data showcase that you can download from there as well. So I think they are live, and we can provide some links to people after this call. So there's a question, Will, around merging of data. So I know this is something that's come up previously around researchers who've got their own exome data, for example, that they've obtained in their own research studies elsewhere. How would they kind of merge that with the biobank data on the platform?
Right. So we recognize that that's a huge use case, and I'm glad to have that question. Sheldon has put together pretty specific documentation on exactly how merging your own exome data and some best practices for doing that with all of our tools. Again, all of our methods are open source. And reading through that documentation, there are directions on how you can take your own data, your own exome data, map it the same way, call the same variants, and aggregate it with the UK Biobank data just as we did. I think this is a really exciting opportunity on the UK Biobank wrap. So if people have questions or want to discuss this, again, please direct that to the community forums.
Thanks, Will. Perhaps if there's time for one last question, there was one that's come through around UTRs and whether UTRs are all in the buffer regions or are some UTR sequences in the target regions. I'm not sure if you can provide some thoughts on that.
Yeah, some of the UTRs are covered in the target. The bed file that we used is available as part of the release. I would just encourage individuals to go grab that bed file and look through and see if what they're interested in is covered in the target or buffer. And again, any questions, we can dig into those details and are happy to do so on the forums. But yes, there are some UTRs covered in the target regions.
Excellent. Well, have you got time for one last question around coverage? So there's a query around, you've expressed the coverage is greater than 20 times coverage for 98% of the bases. I guess, what's the mean coverage? Do you have a sense of that from the work done to date?
I'll probably get yelled at, but the mean coverage for these data is between 50 and 60x. However, Those exact numbers can be found from the data themselves. That's not really a metric that we focus on in our sequencing because we're going for consistency and hitting this 20x. The mean coverage we don't feel is as indicative, but it is somewhere between 50 and 60x.
Okay, excellent, thank you. So I think it's probably the time we've got for questions now. If there's other questions that are coming through, we can certainly look to make sure they are answered either offline or via the community forum after this call. So Will, thanks very much for your time. So perhaps we can move on to the next section, which is Lauren Carson, our Head of Research Development, to talk around applying for access to biobank data.
Thank you, Mark. Good morning or afternoon, depending on where you are in the world. As Mark's already introduced me, next slide, please. So firstly, I just wanted to touch on a few key principles that we have with regards to access at UK Biobank. So data is made available to all bona fide researchers for all types of healthcare related research which is for the public interest. We have no preferential or exclusive access to our data, and the use of the resource is based on, has the same basis whether you're from an academic or commercial background. Next section, please. Researchers only have to pay for the costs of using the resource, and access to any biological samples are limited as our samples are depletable and are therefore carefully controlled and coordinated. And any researchers using our data and undertaking analyses on our data are required to publish their findings and return the results of their findings to us so that other researchers can use them in future. And this includes data from sample analyses as well as imaging data, etc. Next slide, please. So there are three key stages with regards to accessing UK Biobank data. We have registering with the resource, applying for access, and then managing your project. And I'll go through each one of these stages in turn. Next, please. So firstly, registering to use UK Biobank data. Every researcher who uses Biobank data needs to register with us. And to do that, we ask you to go to the access to AMS, which is our access management software, and it can be accessed on this web link here. When you enter this web link, you'll be asked to fill out a registration online form, and within this we ask for you to provide an up-to-date CV or resume, along with a link to your personal profile at the institute that you're applying from. Where possible, we also ask you to provide us with some PubMed references, as well as an email account associated with you from the institute that you're applying from. We then ask you to submit your registration, and each registration application is reviewed personally by our access team and is approved accordingly. Our access team may contact you if there are discrepancies on your registration forms, etc. So please work with us when we ask you to amend anything on your registration form so that we can approve you as quickly as possible. Next, please. So when you have completed your registration steps, you're able to then put in a new application if that's what you wish to do. So the first step would be to go into the AMS system and complete an application form and select what tier of data you wish to use. I'll talk about the tiering system in a moment. The second stage is to then add the researchers who you would like to work on the project with you. If you have any collaborators, and add them as a collaborator. They also need to be approved registered researchers and go through the registration process as well. Thirdly, we ask you to add contact information for a signatory who is authorised to sign the material transfer agreement, or MTA, on behalf of each institute listed on the application. And then we have a few stages internally in which we review your application, and we will provide you with feedback, etc., during this process as well. Once your project has been, your application has been approved, an access fee request will be raised, and the MTA will be sent out to you and your authorised signatories to sign. Following payment of the access fee and return of the signed MTAs, the Access team will then inform you when your data has been released and when your data will be available for access. Next slide, please. So as I previously mentioned, there is a tiering system through which researchers can access our data. And we have tiers 1, 2, and 3. Tier 1 details all of our core data. This is questionnaires and physical measurements, linked healthcare data, etc. Tier 2 not only includes the core data, but also data on assays and enhanced measures such as biochemical assays, hematological assays, etc. And tier 3, which I'm sure lots of you are very interested in, is the core data, the assay and enhanced measures data, as well as the large data sets such as whole genome sequencing and whole exome sequencing data. And this is only made available through the RAP, this tier 3 data. So with regards to costings, for 3 years worth of access, you will be charged £3,000, £6,000 or £9,000 respectively, depending on which tier you have applied for. And if you have any additional institutes listed on your application, they will be charged £1,000 each for 3 years of access in addition to the fees for each tier. However, if you are a researcher from a lower middle income country or a student researcher, you are given a reduced fee of £500 for the first 3 years of the project. And we've also made available that all data sets are able to be accessed on the RAP for researchers from low middle income countries or our student researchers. Next slide, please. However, I appreciate that there will be some researchers who want to be added to existing projects. To do this, we still ask you to register and go through the registration process. But then the principal investigator or PI of that project will then need to go into their portal on AMS to add you to the specific applications. However, if you are joining a project which does not currently have is not previously listed on the project, you will have to sign a new MTA to do that. Next slide, please. And finally, the last stage is managing your project. So we ask that you keep your contact details up to date for us, that you submit any extensions to your project as they're required and not after your project has expired. We ask that you provide annual project reports and any annual confirmation forms as required by the standards set in your MTAs. That you alert us to any publications that you have using our data, and that you adhere to our publication guidelines, which I believe we can put a link to in the chat, and also that you're able to respond quickly to any audit requests UK Biobank send to you, and that you return your results once you finish your project so that we can allow other researchers to use them in future. Next slide, please. So just very quickly at the end, I just wanted to mention a few key hints and tips how to minimise your application time. So firstly, ensure that your legal team has seen our standard MTA template at the point of applying, and we can provide that to you. And I believe we might be able to even put a copy of it in the chat or a link to it in the chat. We are also able to, we also ask that you amend your application in response to our feedback as soon as you can, that you pay your access fee online through AMS. However, if you are having any issues with that, we ask that you firstly liaise with your finance departments to see if they're able to help you resolve these issues to ensure prompt payment, and that you sign the MTA when we send it to you and your authorised signatory as soon as possible. We are now using the DocuSign system, which makes it very quick and easy for you to complete. But that's usually the key stumbling block for researchers applying for our data is delays in signing MTAs. If you ever have any questions or concerns about any of these steps, please feel free to contact us through the AMS system. We have a messaging service through there, or by contacting the Access team at access@ukbiobank.ac.uk. I hope that was clear for all of you, and I'll feel free to answer any questions you may have.
So if we look at some of the Q&A coming through, perhaps the first one, could you just touch on, are there any fee reductions available to researchers?
Yes. So as I've previously mentioned, if you are a researcher from a low middle income country or a student researcher, we offer a reduced rate of £500 for 3 years of access. However, do keep an eye out for the AWS researcher grants, which should be made available fairly soon. This is a grant to offset some of the charges incurred through using the RAP system, so it helps with analysis and storage as well for early career researchers, students, and researchers from low middle income countries as well.
Thanks, Lauren. So there was a query around typical approval time. So perhaps you can talk on how long does it typically take from the point of application submission through to approval?
So at the point that an application is submitted to us to the point of approval currently is a 17-week turnaround. However, on average, about 10 weeks worth of that is us waiting for signed MTAs to come back to us. So as I said at the end, the best thing you can do is warn your legal departments that you've got MTAs coming and prep them with the standard MTA so that they can review it prior to a DocuSign request coming into their inbox.
I think it's worth saying we've done quite a bit of work on the biobank side to try and streamline our processes, and I think that kind of signposting and early warning to your internal departments is really helpful to try and smooth that process, as Lauren says. There's a question around collaborators, and do collaborating institutions have to go through the same kind of application process as the PI? So perhaps you can touch on how do collaborators get added to an application?
So collaborators can be added to the same application. They just have to go through the same registration processes. And then, so they have to register with us, become an approved researcher, and then the PI can add them into the existing projects that are currently running. And then obviously if they're a collaborator from an institute that's not currently listed on the original project, they will need to then go through and sign a new MTA as well.
Okay, I'm just seeing if there's other questions coming through. Caroline, are any of the questions that have come through?
There is one about how to obtain an updated MTA. Yeah, so I will drop in the chat a link to where there is a blank MTA, so you can download one to see that ahead of time. And then the other question is around if they already have approval for the whole exome, how do they reapply for the 300 exome?
Yes, so anybody who's got an ongoing project and would like access to the 300k exome data and any subsequent data through the research analysis platform, please drop us an email or send us a message through AMS, letting us know that you would like to move to the new MTA and pay the new fees and access that data through the platform. I'm sure if you're on an ongoing project, you've had emails from us recently advertising this, and there has been a good deal of demand. So we can't promise that we will get to you immediately. We're working through them as quickly as we can, but just send us an email to access@ukbiobank.ac.uk or send us a message in AMS through your project, and we'll get back to you as soon as we can.
Brilliant. Thank you, Ben.
Thanks, Ben. So in the interest of time, what I suggest we do is we go on to the next session, which is about getting onto the RAP. And then if we have time at the end, we can pick up some of the additional questions that are coming through. So if I stop screen sharing and hand over to you, Anastasia.
Thank you. Okay, so hello, good evening or good morning. My name is Anastasia Sedlakova, and I will make a walkthrough how can you get to UKB research analysis platform and how data are organized there. So what is a sign-up procedure? So Lauren was talking about how you can apply via IMS and make MTA signs. So once you do it, you can sign up into UKB research analysis platform, which is hosted by DNA Nexus. So you do it as ukbiobank.dnanexus.com. And then you need to connect your research analysis platform account to AMS. And I would like to note that your AMS login does not need to match your research analysis platform account login. And so once you do that, you can go to the RAP, and then you can create a project and make your analysis. So in the platform, in the UKB RAP, there are different ways how you can interact with it. So first, there is a UK BRAP UI when you can organize your files, and you can also look at the bulk files. We will talk about it in a few moments. And you can also look in the cohort browser, where you can create cohort and you can explore data set. It could be mainly used for phenotype data. Then you can also use apps, which is basically packed software. For here, I have an example of Swiss Army Knife, which is a software which contains different bioinformatics tools. Or you can interact with UK BRAP using two types of Jupyter Lab. One is a single node Jupyter Lab, where you can use, for example, you can program in Stata, or you can use machine learning settings if you want to make some heavy analysis, for example, image processing. Or you can use our Spark Jupyter Lab if you want to get the data. I will talk about what kind of data do we have in a moment. I just want to have an overview. What do we have? So we have a UK BRAP community. So once you get the presentation, you can go through all of the links that we have. We have a quick start section on our documentation when you can go through different tutorials, which can help you to access, to learn quickly how to work in UK BRAP. There's also a community forum where you can ask questions. We have our newsletter, and we also have our public GitHub repository where we are publishing some Jupyter notebooks that can help you to start using UK BRAP. And you can also contact us at community@dnanexus.com. I will also like, if you were interested in what are some Jupyter labs or Jupyter notebooks, then I would definitely recommend you to go to our Jupyter notebook training, or you can go to our UK BRAP overview, where we'll cover some of the things that I will show here, but in more details. And also you can make an exploring and analysis UK Biobank data with Jupyter notebooks, so something even more in depth. So, okay, now I will go into the different steps how you can create your project, and then I will show you what are the data organization on UK BRAP. So once you registered in your MS, you get approved, then you can sign in UK BRAP and link your account. So once you log into your UK BRAP, you will see that you have no projects, it's empty. So you can create a new project. You click on new project, you select your application ID. You can have like multiple application IDs, so you can put here like the appropriate one. And then you will see that there is, so you see here that there is a new project appeared, but you see that the status is dispensing. So that means that the data are dispensed currently to you based on your application, and you will have to wait about 1 or 2 hours, or maybe longer if there will be like a bigger queue. So I would recommend you to start it before, for example, like a day before, if you want to make your analysis. So this was about how you can start your project. And once you open your project, the data will be organized in 2 different types. So one would be data set, and one would be bulk data. So data set contains phenotype information and genotype information, and you can access it using Spark applications, for example, Jupyter Lab, Spark Jupyter Lab, or you can access it using cohort browser, which is more like graphical user interface, where you can go field by field and you can show, and you can look at the data. I will show it to you in a moment. We also have bulk data. So files are associated with participants. So for example, imaging data or genotype data, and they can be accessed and processed via apps, or you can also process them in Jupyter notebooks. So if we can briefly look at the structure of UKB data on REP. So there is a data set, then the data set contains entity. We could say that the entity is equivalent to a table, and then each entity has multiple fields, and the fields are equivalent to a column in the table. But they also could have some associated metadata. For example, if we have a field which is weight, we could have also metadata as units, and also link to UK Biobank documentation. For a bulk data files, there could be, as I said, like imaging or genotype files, and each of the file could have field ID if it's per participant, if field ID or EID if it's per participant. You can search using field ID or EID, and you can use these properties for your further analysis. If we look a bit more into depth, how the bulk files are organized, you could see that there are multiple folders here. So basically, samples are organized by first 2 digits. So here we have folder 10, and all of the samples that are there are starting, their EIDs are starting with 10. So what I also want you to show, just briefly, that as I said, that the data set could be accessed using either Spark JupyterLab, or it could be accessed using Cohort Browser. So if you are just now, you applied for UK BRAP and you're just freshly starting with your project, I really recommend you to go to Cohort Browser and to look at the data, so get to know the data or get inspired. So how can you do that? Here we see that there is some project that I have, and in the root directory, meaning once you open the project, you can see either dataset or cohort. So here are already created something, so there are different cohorts, but you will see just dataset. You click on that, and in the new tab, you will see Cohort Browser. So Cohort Browser can enable you to look at to create cohorts. For example, here I have male and female samples, also to compare cohorts and to visualize data, to explore them. So there is a plenty of opportunities how you can start easily to use UK BRAP. Yeah. Yes, so, and I think that we have time for questions now.
I think we've got time for a few questions. I think there's a question around how often the phenotype data is updated in the RAP. And when it is updated, how do researchers request it? Does it get redispensed or not? I don't know, Anastasia, whether you want to take that one.
So I can answer partly on that. So so I know that we're working on automatical redispensing the data. So the moment there is any updates, so the data are redispensed automatically. I just not sure if we already have it deployed, but we're working toward that. But I think that maybe Mark, you can answer about the phenotype of data.
Yeah, I can answer the first part. So the intent is that the data in RAP will reflect what's available within Showcase. There will be a short delay. Typically, when data is being published to Showcase, it will be a short period after when it becomes available in the RAP. And as Anastasia says, right now, when it has been updated in RAP, you will need to do a redispense at this time, but the functionality is coming in due course for that redispense into an existing application to be automatic. So that will be appearing in due course. There's a question around, is the RAP optional for researchers to analyze the data? And the answer to that is yes, it is optional. Researchers can still continue to download the majority of biobank data from Showcase if they so wish. We are looking to make the platform very functionally rich and make it a place where researchers want to come and use out of preference. But there is one point to affirm that the new WES data, so the 300K exome data, the forthcoming 450K exome data at the end of October, and then the 200K whole genome data will only be available within the RAP, and they will not be available for download from Showcase. And that is a change that we communicated earlier this year. And then again, there's been a couple of questions around, is that only for the CRAMs? Can researchers download the PBCFs? And again, just to provide some clarity to that, it's both the CRAMs, the VCFs, the PBCFs, and the BGen and Blink files are only accessible for analysis within the platform, and they will not be downloadable from the RAP environment. So hopefully that's clarified that. There's a question on information on cost estimations. I'm not sure, Anastasia, whether there is information on the kind of the rate card available within the RAP, but I'm not sure if there's any particular thoughts on that we can provide.
Yeah, I think that maybe I will ask my colleague Maria if she can send to chat a link to our documentation where we have some billing information.
And again, feedback like that is, I think, going to be really useful within the new community portal. I think that is now in place and live, and we'd encourage everybody to sign up to the community portal and to be kind of posing these kind of questions and sharing their own kind of learnings and best practice. And when you're undertaking certain kinds of assays, again, some of the kind of costing sides, I'm sure will be helpful to other researchers as well. So please do encourage you to make use of the portal environment. There's a question about whether AMS provides facility for non-researchers to access, for example, to help finance departments to facilitate the online payment fees. And that isn't possible today. But again, I think there are a number of ways that researchers can communicate with the Access team to arrange for that to be done offline, or researchers, if they're able to do so, can do that themselves through the AMS portal directly. But right now it is only available to registered researchers to be able to make those payments through the portal itself. So just looking at the questions coming through. Question about, can we export summary data or data results from RAP for downstream analysis? And the answer to that is yes. So whilst you can't download things like the VCFs and PVCFs, any summary stats that you generate from your analyses can be downloaded out of the platform for use in your own environments if you so choose. There's a question, perhaps Anastasia, you could touch on this. So there's a question about if a researcher is using Jupyter notebooks or running any apps within RAP, just how does that kind of charging mechanism work in terms of what is it a researcher will need to pay for?
So a researcher is starting with £40 on UKB RAP. The researcher can try to work, for example, run Jupyter notebooks. So when you're working in Jupyter notebooks, when you're launching it, and if you want to have more details, we really encourage you to come to our webinar. You are so there is some cost estimation based on what parameters you selected, and then if you increase duration of your Jupyter Lab, then the cost increase, or if you terminated earlier, then the cost is decreased. So it's computed like that. And yeah, again, we will encourage you to look at the links that Maria sent to the chat and to go through the billing information that we have.
So it's a really good question about can researchers install their own tools within the platform? And if so, who will be able to see and use these tools? So I think that's a really good question. Perhaps you can just talk to how that kind of works.
Yeah, so we have apps, which is the software that we are supporting, and then there are applets that the researchers can create their own like apps. And then I'm not sure what is with the sharing, but definitely that's possible that you can bring your own tools and then you can run it as an applet. And maybe if Maria, you can send some information to the chat about how to create your own applet. We have a documentation about that.
There's a question about large-scale assays that might come downstream, what might fall under tier 3. Perhaps, I don't know, Lauren, if you want to take that one, or I'm happy to answer that. It's in the interest of time. It's so right now under tier 3, I think there's a graphic that Lauren shared in her slides, and these slides will be available after this session. So right now, tier 3 is for imaging data, and then the sequencing data, both whole exome and whole genome. But in future, there may be additional data sets added to tier 3. So for example, there's been a lot of interest in in mass spectrometry based proteomics and metabolomics, and it may well be that they would get added to tier 3 in due course, given the quite comprehensive size of those data sets. So again, that's something that we'll be looking at over time as new enhancements are pushed forward, and as new data types are generated. There will be a process that we go through to look at which tier they should sit in, and we'll be communicating that to researchers downstream. Now, there are questions that have been asked which we've not had a chance to get to. and we will make sure that they are answered after this webinar directly with the people who've asked them and via the community portal. I wonder, Asha, if I hand back to you, if there's anything you want to say in terms of wrapping up.
Absolutely. Thank you so much again for everyone for joining and for all the activity in the question, the Q&A section too. And I really just want to sort of encourage everyone, as we sort of talked about, to sign up for the community forum where a lot of these questions are being posted and also answered, as well as a number of other questions too. Also, maybe just in wrapping up, want to just emphasize some of the other webinars that are happening. So October 12th, as we talked about, there's going to be a UKB wrap overview. We encourage you to go to the community forum and to the website to look at that and to sign up for that. And also, as mentioned as well too, we have a Jupyter Notebooks webinar also happening on October 18th. We also welcome any suggestions for additional webinars or education training and documentation that you'd like to see in support of your activity on RAP, and also just with the UK Biobank data. We really appreciate all of your engagement. And again, just wanted to thank everyone for joining today. I want to thank the panelists for providing such great information, for answering the questions. And again, thanks to everyone simply for being a part of this journey.